Now we're recording. Hi, everyone. I'm here with Jasper Götting. Welcome to the ACIT Science Podcast. I'm just going to start off with a couple of words on your background. You're currently doing your PhD at the Institute of Virology of the Hannover Medical School, focusing on the sequencing and monitoring of viruses. We are going to talk about virology, sequencing, variants, and also some relationships to the current pandemic and future pandemic risks. But your research topic also nicely relates to the second big topic that we're going to discuss today during our conversation, which is your work or engagement in the context of the effective altruism community. And for you personally, these topics are nicely related since you actually chose your career path out of reasons close to the heart of effective altruism, which are thinking about the long-term future of mankind and certain existential risks associated with it, like biorisks, and also the general question of how to actually make most use of your time to do most good in the world. And finally,
Yes.
you. Yeah. It was a
The
very resounding
latency
yes. Yeah.
latency is intense. Yeah.
Yeah, and finally, you're also involved in classic EA topics such as effective giving and animal welfare and many other topics. So I'm sure we will have a lot of things to talk about. So let's just dive right in with a small icebreaker question. What is a virus actually? What do we mean by it?
Yes. Thank you. First of all, thanks for being here. Really looking forward to our conversation. So, what is a virus? To um, to make it very brief, a virus is a sort of self-replicating biological machine. And it's kind of the most minimal self-replicating biological machine, but with a very clear distinction that a virus is a parasite, so it needs a host to replicate. And viruses are in essence small packages of genetic information, so either DNA or RNA, which are encapsulated in some sort of shell, which can be just a protein shell, or it can be a protein shell with also a lipid membrane around them. And viruses enter and infect cells, hijack their machinery for replication, and then essentially use this cell to make a bunch of copies of themselves and spread them. And there's a bit of a debate whether you can count a virus as like alive or not. I think most people would agree that viruses are in fact not alive. But others draw the distinction between virions, so the actual tiny particles which go into cells, but say that maybe a virus itself is the whole life cycle of the virion and the infected cell, which would then be like the virus factory, some sort of second life cycle, and might argue that the life cycle as a whole is alive. But yeah, in short, tiny self-replicating parasites which just make copies of themselves using other cells.
Yeah, so it's probably a question of terminology. Whether it's alive kind of depends on. So it's not doing any like energy householding, any. It can't upkeep its like it doesn't do any homeostasis to keep itself up, but it has to rely on a cell to do all the relevant work for it, basically.
Yeah, that's correct. It's obligatory parasite.
But in the same way, viruses wouldn't be here without any life on Earth. So they kind of fit to or they are really well adjusted to the shape of life on Earth. So in a sense, they really are kind of part of life on Earth.
Oh yeah, of course. And they probably originated by genes from already existing cells, making themselves mobile and independent, so to speak. Like single genes escaping from a cell by essentially producing their own hull and their own shell and going their own path, mainly a path into other cells and doing more of the replication. But the origin of viruses is a bit of a bit of a mystery. There are very good theories, but I don't think it's ultimately resolved yet.
So at the heart of the genetic machinery of life, we actually have DNA and RNA, and viruses also consist of the same basic genetic structure. So maybe we can use this opportunity to distinguish between DNA viruses and RNA viruses, maybe also connect it a little bit to what function DNA and RNA play in the human body.
Yeah, of course. I know you already had previous podcasts, and your listeners are probably somewhat knowledgeable in those topics already. But as a brief recap, um, our organism uses DNA as a template for storing the genetic information, and usually uses RNA to transcribe certain parts, certain genes, certain proteins it needs as RNA, or usually messenger RNA, which then get translated at the ribosome, like some sort of cellular assembly machinery, into the final protein, the amino acid based tiny machines, which actually do all the work in the body and also constitute most of the structures. And DNA is DNA and RNA differ by an oxygen group essentially. One is a desoxyribonucleic acid, the other is a ribonucleic acid, and this one oxygen group makes RNA less stable since it's able to degrade itself in an autocatalytic way. And in our normal cellular machinery, this is also like part of the natural process that RNA degrades relatively fast. It's just used as a template, which briefly brings this information, this gene, which needs to be transcribed and translated to the ribosome, where it then gets produced, and the RNA is eventually degraded. For viruses, the RNA DNA distinction is functional. Like, they ultimately evolved very diverse pathways of reproducing, which include different locations in the cell where viruses reproduce, be it in the nucleus where the host DNA is, or outside in the cytoplasm near the ribosomes. Of course, if you have an RNA virus, the genome can often directly go to ribosome and produce proteins, since the virus genome itself is a sort of messenger RNA, whereas DNA viruses need to first make RNA from their DNA genome, mostly using the host machinery, which then get produced to proteins. So they mainly fill different evolutionary niches by just having DNA viruses, having RNA viruses, and one relatively major difference is the rate of evolution, since DNA replication is usually included with proofreading. So the DNA polymerases, which amplify the DNA, they have a proofreading function, which checks whether an inserted base like ACTG is correct, and corresponds to the template base on the opposite strand of the DNA. And if that's not the case, the base gets just kicked out and the correct base is placed in. Whereas RNA polymerases usually don't have a proofreading function. Big exception are actually coronaviruses. So the SARS-CoV-2 virus has a proofreading polymerase, which leads to a reduced rate of evolution. But many viruses, like the HI virus, which causes AIDS, they have an immensely high rate of evolution, since they don't have any proofreading. So errors really stack up quickly, which makes, of course, vaccinations and control of those viruses very difficult, as we see with HIV.
Yeah, I read recently that the rate of mutation of the RNA viruses is like approximately 100 times larger than DNA viruses. I don't know if you can generalize that so nicely, but that basically the rate of mutation is so fast, like it's on the edge of error correction, basically. So if it would be faster, the virus doesn't work anymore. So it's really very rapidly evolving.
Yeah, that's correct, and it's quite interesting. Like you said, many RNA viruses really do evolve at the error thresholds, and if you somehow can interfere with the replication processes and increase the error rate, the virus population actually collapses since the error rate is too high, and functional virions are actually not produced anymore, or not produced in a high enough amount, since there are too many errors. And some antivirals actually work in that way. They introduce errors in the replication process, which pushes a virus population over this error threshold into the error catastrophe, as it's very aptly named.
Does it make sense to say that RNA viruses are better at adjusting themselves to like shifting environments, or because they are evolving so rapidly? Does it give them an evolutionary advantage to DNA viruses, or does it kind of correlate to where they pop up?
Yes, like generally, I would say they are quicker to adapt. But then again, RNA viruses are usually smaller, and DNA viruses, with their larger genomes, often have interesting ways to, for example, evade host immunity while still having a somewhat lower rate of evolution. So essentially, all viruses you see out there which have diseases named after them are evolutionary quite fit. Otherwise, we wouldn't still have them around after tens or hundreds or even millions of years. But they definitely have different approaches of evading the selection pressure given by our immune systems.
So you already mentioned HIV. So I think some of the viruses that interest us most are RNA viruses, like especially currently SARS-CoV-2, but also the influenza viruses and HIV. I think, are there any others?
Yes, definitely. For example, the hepatitis viruses are RNA viruses. For example, yellow fever virus is an RNA virus. Yeah, a lot of for example, the arboviruses, like Zika, which also was quite popular a couple of years ago. Yeah, there are also RNA viruses, but it's not like we don't have our fair share of problems with DNA viruses. Like the herpes viruses are not as high in like pandemic potential, but definitely definitely have a a total high disease burden on humanity, as well as adenoviruses, and also the pox virus, which caused smallpox, which is fortunately already extinct, was also a DNA virus.
And probably the deadliest virus in history, with like
Yes,
700
exactly.
million people killed during the course of humanity. Yeah, interesting. At which group of viruses do you think holds like the largest pandemic potential? The coronaviruses or the influenza viruses?
Um, it's a bit icky to talk about that, since if you think about or talk about biological risks and potentially catastrophic biological risks, the issue of info hazards or bioinfo hazards comes up, like the idea that some knowledge is inherently risky. And I'm not saying that like discussing the pandemic potential of certain virus families falls into that category, but it's quite near, since you generally don't want to give give good ideas to bad people, to say it very provocatively. Um, I think the biosecurity community, which thinks about catastrophic risk, is more on the side of not sharing knowledge widely. So I probably wouldn't discuss like specific pandemic potential of certain virus families in closer detail.
Yeah,
And you can think about whether you want to edit this out or not. But generally,
no,
yes,
no. I think that's a
the
very, very important comment
yeah.
or insight.
The influenza viruses are definitely out there as having a high potential for big pandemics, as we saw over and over again, essentially, be it the flu of 1918, and also larger flus in the 70s, which people don't remember that well, but which had like a high number of infected and also a high, not a high case fatality, but overall a lot of deaths.
Yeah. And talking about influenza viruses, because I also wanted to transition to that because it's quite interesting how they work and that there's this mechanism within their, like the way they shuffle their DNA around. So,
Yeah, that's quite interesting. Influenza is a segmented virus, so there's not like a single RNA strand in the virus capsid, which then gets turned into proteins. But they have 8 segments, and each segment has like different genes or a different gene on it, which codes for a different protein. And when they infect, when multiple or two different types infect a single host, they can essentially shuffle around their segments, which then get repackaged into new virions. And this shuffling, this genetic shift, leads to quite rapid evolution, since essentially you swap out whole genes for different versions, which might not have been encountered by the immune system of a certain species, for example ours, which makes it quite challenging to develop a vaccine. And that's kind of the whole thing about the yearly gamble with the flu vaccine, whether it will work well against the forecast version and or not, and which also makes the generation of like a universal flu vaccine quite tricky. But there are of course groups working hard on that to find some feature of the virus proteins which are conserved um, over all segments, over all possible variants, which would then work as a vaccine and as a target.
I heard about a similar idea in the context of coronaviruses that you can basically find a vaccine for all the SARS viruses, for example, by just finding this common feature in the spike protein or something. Do you know more details about this?
Yeah. So the tricky thing in general is that viruses, of course, evolve, and as soon as you try to use one part of the protein as a target to, for example, immunize against the virus, you inadvertently create a selective pressure, so that any virus variant which is not targeted by this vaccine or by this antibody has a advantage in replicating. Um, but there are, of course, in many proteins regions which are like central for the function or for the structure of the protein, which cannot be changed. For example, you need a certain amino acid in this position for the receptor to work, or for the protein to dock, or for the enzyme to catalyze a reaction. And if you are able to find such regions and find such a target, then it is possible to create like an unavoidable antibody, for example, since the virus doesn't have a chance to evolve around that, since it can't change the protein in that position.
And are we potentially facing a similar challenge with the current coronavirus pandemic that basically the virus is evolving and constantly you have new variants that evade the immune system, or you think this these also properties of the virus themselves, like this error coding mechanism that basically puts a lid on the rate of mutation, and the differences with respect to the influenza viruses that actually have this shuffling mechanism gives us some hope that we won't run into these issues.
I don't know how much of the possible sequence space the current coronavirus already has explored. There are certainly more variants out there, like more potential variants, which are very likely higher on the fitness landscape, on the theoretical fitness landscape, with its peaks of high um, replication. Yeah, I'm not sure how large the sequence diversity out there still is, and how fit, for example, the Delta variants, which we now see predominantly, how much fitter the virus could still be. But we, of course, run into the issue of constantly appearing variants which might be able to evade immunity, given that we still have, for example, here in Germany, around like 60% people vaccinated, which is not enough for herd immunity. And if we then look into other countries where the vaccination rate is much lower, then we very much can imagine that we will see a constant trickle of variants, which might be uh, might be able to say just infect like 5 to 10% of already either vaccinated or converted, like previously sick, but then genesen. What's
Recovered.
recovered? Yes, thanks. But recovered patients. Um, but yeah, not 100% sure about that. There are people out there which probably thought way longer about that, which are able to answer that more, more definitely.
Okay, I'm not sure if you know anything about that, but I was just wondering, like in this mutation space, we have a lot of coronaviruses, or like I think 4 or 5 coronaviruses already just being part of the daily or yearly ritual of having a minor cold. So they are also probably still mutating, but apparently there's no real risk of them mutating to become really dangerous again. So it's just because they don't have as much expressive range when it comes to mutation, or does that risk actually exist? Or I don't know if you know anything about that.
Not 100% sure. So they probably started out very similar to SARS-CoV-2 now. So initially quite harsh, or at least harsher than they currently are, because I don't think anybody even remotely unhealthy is dying of the common cold, unless you have some really weak immune system or pre-existing health conditions. But like over long times, over many generations, viruses tend to get milder, or zoonoses tend to get milder when introduced in the human population and remaining there. Mechanistically, I'm not 100% sure why this is the case. There is, of course, the trade-off between like virulence and transmissibility. The more virulent the virus is, the more likely it kills its host, which would then lead to the host not transmitting the virus. So there is some evolutionary pressure to viruses which are able to infect you like often and maybe for some time, but not kill you, but making you like spread them a lot by sneezing and coughing and so on and so forth. So this might be the main driver for this trend. We see that they just get milder because they essentially replicate best and spread best when just making you cough and sneeze, but not much else.
Yeah, yeah. I think that it always sounds reassuring, but there's probably no way of guaranteeing, like, from a scientific perspective, like just guaranteeing that they won't mutate in a way. But it's probably what we have observed so far in the last couple of years or centuries, even.
Yeah, I wouldn't worry about cold viruses suddenly getting very very dangerous or very virulent. I think there are currently just not there are no selective pressures on them for becoming more dangerous. Um, yeah.
And one interesting point about herd immunity that I remember reading about at one point is that basically the 1918 flu, or with the flu, you also have these different strands of the AHN virus that causes the flu. And then basically, if children get sick, the risk of getting seriously ill is usually lower than for old people. And the real problem with these strands was that basically in 1918 they occurred for the first time and there was no basic immunity within the certain parts of the population for this virus. And I think there was like some pandemic in 1880 with a similar flu virus, so that's why like middle-aged people weren't affected too much, but like the 20 to 30 generation of young people had never seen that specific flu virus before that, so that's why they were hit really hard. Sometimes these immunities interact in unexpected ways.
Yeah, there was I think it was called the Russian flu in the 1880s.
Yeah, yeah.
So some non-existing immunity definitely played a role there. Yeah.
So you mentioned people with very frail or weak immune systems, and this connects to the research you are doing. Maybe we can start by talking about what research you're actually doing and what you're focusing on and how this connects to like application in the context of organ transplants and people with very weak immune systems.
Yes. So my research in the Institute of Virology at the Hannover Medical School mainly deals with the diversity and evolution of DNA viruses. The Hannover Medical School is a transplant center. They do a lot of transplants of various organs, but also, for example, bone marrow for example, children with leukemia. And during transplants, patients usually get immunosuppressed, so their immune system doesn't reject the donated organ. And while being immunosuppressed, a lot of the normal viruses which everybody has, like for example herpes viruses, which are not really a problem for anybody except maybe mild annoyance, they do become very dangerous since this constant like inactivating pressure of the immune system is suddenly missing, and they reactivate from their latency and can essentially replicate freely in the body and cause some serious disease either in certain organs or throughout the whole body, which can lead from rejection of the donated organ to like disseminated disease in multiple organs to multi-organ failure and subsequently death. And while this is now quite well managed in the hospitals and in the transplant setting with prophylactic treatment with antivirals, there was still the question whether some viral variants are maybe more dangerous for transplant patients than others. So my research dealt with essentially sampling, sequencing, and then looking at the diversity of cytomegalovirus, which is one of the herpes viruses. Yeah, period. That's that's pretty much the first big project of my PhD. Maybe we can talk more about that before I go on to further projects.
Yeah, maybe we can. I think it's very interesting to stress that point again that we are already infected by viruses that we don't really notice in everyday life. I think because herpes is so famous, but I think really misunderstood because it always seems like you can get herpes by um, especially sexual encounters, but a lot of people already have herpes, maybe even since their youth or young days. So
Yeah. Usually since their youth, actually, or even since infancy. Like herpes viruses are quite diverse. We have 8 different ones.
Herpes
And
simplex.
virus 1, the classical herpes virus, which makes the lesions on the lips, is just one of 8 herpes viruses. Herpes simplex 2 can also do lesions, but usually not on the mouth, but on the genitalia. But there's also, for example, VZV, varicella zoster virus, which is the causative agent of chickenpox and shingles. And for example, human cytomegalovirus, which I was researching, which doesn't really do like some charismatic disease which you can really easily identify, but it's usually just like a mild cold during infancy. It often gets transmitted via breast milk during during infancy, and that's it. Then you have cytomegalovirus, and should you ever have a child and breastfeed it, then that child probably also gets cytomegalovirus since it reactivates in the mame. So yeah, we do have quite a diverse repertoire of viruses latent in us, often since childhood. But they also usually don't worry us too much since they're just there. And if we're stressed and our immune system is maybe a bit weak, then they might reactivate and we get like lesions on the lips or a bit cold or something like that. But it's not a big deal as long as the immune system is healthy.
So that means that a lot of these can actually be. So there's basically this constant struggle between the immune system and these latent viruses. So it's always suppressing them kind of actively. And when you get stressed or when you are weakened by some kind of reason and your immune system is occupied with other things, then they just basically grab the opportunity to get stronger again. And that causes the symptoms.
Yeah, essentially, yes. So they are very specialized in their latency. Like it's not any virus could become latent just by experiencing constant pressure from the immune system. So they have a quite complicated machinery which allows them to essentially become like silent mini chromosomes alongside your actual big human chromosomes, and be treated quite similarly by the human replication machinery. So they're a bit more sophisticated in their latency than just constantly repressed by the immune system. But it kind of boils down to that, yes. So if your immune system suddenly is weakened or just completely gone due to some immunosuppression during a transplant, they probably stochastically reactivate, and if they don't encounter any pressure, any force, then they just replicate. And completely free virus replication is never good for any tissue since the viruses just destroy everything.
Yeah, but in a way, it's also not good for the virus because it's destroying the host too much.
Yeah, that's true. But evolutionary speaking, immunosuppressed patients during lung transplant are not the most natural thing in the world either, and it mostly had to deal with infants with a healthy immune system over the millions of years of hominid evolution.
So now, during the COVID pandemic, we're starting to be very familiar with the concept of monitoring viruses and monitoring how they are, or even the whole concept of mutants and different versions of the same virus. So is monitoring viruses important in general, and why were you involved in this project in the context of the herpes viruses? And what are the kind of scientific promises, and what are you hoping to find out?
Yes, so monitoring viruses in general, I'm not sure whether I even have to to make the pitch for that during the current times, and it's of course immensely useful for humanity as a whole to know what's out there what viruses we have to look out for. And in a hospital setting where you are transplanting organs, you obviously have to know whether the donor of an organ has, for example, cytomegalovirus, and whether the recipient doesn't have it. So you would have to be prepared for some sort of cytomegalovirus reactivation upon transplantation, especially since it affects the outcome so much. But the idea behind the project was to find variants which increase virulence or mortality or general outcome of in the transplant setting. That was part of the first study, where we found out that the viral diversity is not really deterministic for transplant outcome. So it's mostly driven by host immune factors. Although what's quite striking is that multiple strain infections, so when a person has not just like a single strain of a virus stemming from a single transmission event, but multiple strains present, which come from, say, multiple infections, or one strain they had themselves and another strain coming from a donor organ, for example. That those are generally more dangerous, since they, when reactivating, just have a much higher viral load. So many more viruses in the blood, in the organs, since the latent repertoire of not just one, but essentially two infections comes up at once, which does worsen the outcome. And the other project relating cytomegalovirus was the transmission in breast milk, where we looked at the breast milk of women who just gave birth to preterm infants. And cytomegalovirus is also quite dangerous for preterm infants as well, since they don't have fully developed body and organs yet. And there we looked whether we see transmission of single versus multiple strains, or transmission of specific genotypes of the virus, preferably into the breast milk, since we had blood and milk. But also here we could see that cytomegalovirus really reactivates broadly during pregnancy and during breastfeeding, which was also quite interesting, since it reactivates only in the breasts, only in the tissue there, and not in the whole body. So it's really evolved to be transmitted via breastfeeding. And we saw that the mother essentially transmits her whole CMV, so cytomegalovirus repertoire, up to 4 or 5 different strains from various infection events at once to the infant during breastfeeding.
Yeah, that's very interesting. Like a very smart way of the virus to take care of
Yeah,
it.
quite ingenious. That's true.
Yeah. Yeah, it's fascinating how well adjusted they seem to be, that the virus implicitly kind of understands how to get into the breast at the right time, so it can nicely move on to the next
Yes,
host.
exactly. Well, to be fair, the virus itself doesn't move into the breast. It's more like the immune cells in which it is latent, which move into the tissue during breastfeeding. But also herpes viruses are quite complex. They are one of the biggest viruses known to infect humans. Not known in general. There was this rise of giant viruses recently, which infect mostly amoebas, which are very, very big. But of the viruses of humans, herpes viruses are one of the biggest, with up to 250,000 base pairs or nucleotides in length, which, if you don't have anything to scale, might not sound that much. Like coronaviruses or SARS-CoV-2 is roughly 30,000 nucleotides in length. A lot of the smaller RNA viruses are around 8,000 to 10,000 nucleotides. And with 250,000 nucleotides, the herpes viruses are really quite massive. And they don't have only 6 or 7 genes like many of the smaller viruses. do, but they have probably over 170 genes. Like, the genomes are not fully annotated yet. Researchers are still discovering new genes in the herpes viruses, but they have at least over 150 different genes involved in quite complex regulation of host immunity and immune evasion and so on and so forth. Yeah, so they're quite complex for just a little particle which hijacks cells.
Yeah, it's quite ingenious. Like, I also wanted to mention that earlier, when it comes to understanding the functions of these also newly arisen viruses like the coronavirus, like how people actually are starting to figure out how these work and which part of the genome serves what kind of function. And with these mutations, for example, people have also been explaining, yeah, this is a mutation in the spike protein that facilitates binding to the receptor, for example. So these kind of ideas, how you can actually decode the genome of a virus and understand where the function actually resides and what kind of mutations could possibly then end up making it more dangerous or less dangerous.
Yeah, that's quite tricky. You're right. That's not an easy task. You can just very easily look for regions in the genome which are likely to produce messenger RNA and subsequently protein, just by looking at a start and a stop codon. So every gene begins, or every protein begins with an ATG, which codes for the amino acid methionine, and ends with one of several stop codons. And if you just look at the DNA strand, you can just look for any region which starts with an ATG and is essentially uninterrupted and eventually ends at a stop codon. And that was often the first step in finding out new genes or new proteins, just by looking for those open reading frames, they are called. But like, they're not the panacea. They're not like foolproof. And we now know that also, like, very small regions which start with an ATG and end with a stop codon are sometimes translated into like very small peptides, so very small chains of amino acids, which just serve some yet unknown regulatory function, but are not like full blown proteins with a complex function, but just serve regulatory purposes. And so it's quite difficult. A lot of the times, it's just a lot of like lab work, wet lab work, experimentation in cell cultures, looking for proteins in various biochemical assays which would fit to certain open reading frames which you discovered and detected on the sequence, and then further confirmation whether that's actually the case. And if you have annotated a genome just by experimentation, you can then look for analogous genes and proteins and open reading frames on other genomes of similar viruses or genomes. I mean, this is of course not restricted to viruses. This is a general research topic: how to find and annotate new proteins, how to discover those. And that's actually quite hard if you suddenly encounter any species which you don't have any well annotated close relatives for, because you essentially have to start from scratch and find out which protein does what.
Yeah. Now, at least with advances like AlphaFold, we have some handle on how to translate genes into proteins in the best case, or to understand how amino acids actually get folded into these larger structures.
Yeah, that's true. And that's very exciting. Like, structural biologists were already quite smart. Like, we knew how to. I say we. I'm not a structural biologist, but they knew how to look for certain like structural elements which might have similar functions. And it's, for example, it's quite easy to detect a helix of amino acids in a DNA sequence, or find and annotate some sort of sheets which might fold into higher degree structures. And if you just have like a long sequence and have a characteristic sequence of helices to sheets, then you can already find analogous proteins in your database, which might have different amino acids at each position, but have a similar overall structure and overall architecture, which might then give you a hint to the actual function of the protein, especially if they then also share a similar active center. So a lot of enzymes have like one part of the enzyme essentially where some reaction takes place and some reactions catalyzed, which is often done by just a small number, like I don't know, 2 to some amino acid which catalyze some complex reaction with their residues. And if that's similar in one protein to the other, then you can also make some guess and some inference about the function if the overall structure is similar, even though the actual amino acids at all the other parts of the protein might be completely different. But yeah, like you said, with AlphaFold and the constantly improving advances in machine learning and AI, and also corresponding increase in data we have, we will probably see this getting more streamlined in the future.
Yeah, I not put down as a more general question how new techniques are changing the field. I was more thinking of CRISPR and these more like wet lab techniques, but as you mentioned, AI and machine learning are also playing their part in changing biology to a pretty impressive degree. So what are like other techniques you can think of, or especially like CRISPR, but also others that are influencing the way biology is done?
I mean, mRNA vaccines are huge right now, and I think very rightly so. They're like not like a classical molecular biology tool, and we can't really say that they will change biology, but they certainly will change biotech and pharma as a way of a programmable vaccine and a programmable countermeasure. Generally, every technique which is programmable in scare quotes is quite potent, since it just allows you to do things which were previously only possible via either very complicated specific design or just very crude techniques. And like you said, CRISPR is huge. You can just target an enzyme very specifically to a very specific sequence with very low off-target effects, and I don't know, either change it or cut it or degrade it or whatever. And that's pretty huge. But yeah, I would say, given the huge amount of biological data that's being generated, I would guess that the very next big revolution is more informatics based. Either like making sense of the data that's already there, or being able to make experiments or set up experiments in a high throughput way, and then automate the analysis of experiments via, be it, for example, some directed evolution experiments where you just get a big robot and pipette tens of thousands of plates of some variation of a virus to see which one is the fittest, and then analyze everything with machine learning or so on. Or protein design, which you can also really scale up if you have automation and machine learning on the analysis side. So that's pretty exciting, but also I would guess, and I'm not really sure what the current advances in this field are, natural language processing. There are huge amounts of research already in databases. And a lot of the time, fields are working on like the same protein or same pathway, but from different directions, but it's never getting really synthesized. And they always tell you, yeah, you have to look over the edge of your plate. You have to look also interdisciplinary and what, as a virologist, for example, what the immunologists are doing. And that's true, but the problem is that like all subfields are like immensely huge, and the databases are like brimming with papers, and it's hard to really dig through that as a human. So some sort of system which would automate and maybe even start to synthesize knowledge in those huge research databases is probably also a gold mine, given how much connections you could draw between published research.
Yeah, that's fascinating that we already are doing so much research that it's basically too much for a human being to make sense of, and then automating that in some kind of way
I guess there are
is
also
actually the way
too
to
little
go.
incentives to just hire a person specifically for for doing reading and interconnection work.
Yeah. Yeah, I think if you just collected all of the bachelor theses that have already been done and no one cared about, then you could probably win a Nobel Prize or
Or
two
burn them and nobody
just out of the
would know
all
the
the.
difference.
Yeah, I mean, I think we sometimes it's yeah, like this general idea of now the internet just being too enormous and becoming more a problem of like how to find information and not like the having the information lying around somewhere. It's more like a searchable space that you need to sift through. And that's why Google is also the biggest tech company, I think, on the planet. They started by searching the web because having the web is not enough. It's actually like a really like interesting perspective. I wanted to also get back to like the programmability part because you mentioned mRNA vaccines are programmable. Just to get into that a little more and compare it how classical vaccines like the polio vaccine or still also the flu vaccines are developed and just highlight the benefits of this programmability.
Yeah. So with the mRNA vaccine and also with the vector-based vaccine, for example, the Johnson & Johnson and the AstraZeneca vaccine, you don't give parts of the virus into like your bloodstream, which would then get recognized and taken up by immune cells and presented, and antibodies would be generated. But you essentially inject the code itself into your cell. Like the mRNA, be it either in a lipid nanoparticle with the Pfizer BioNTech or Moderna vaccine, or the mRNA as part of the adenovirus vector genome, which is in the Johnson & Johnson and AstraZeneca vaccine, your cell gets infected by this either this vector, this adenovirus vector, or by this lipid nanoparticle. And your cell then produces, based on the mRNA or based on the adenovirus mRNA, produces the virus protein itself and expresses the protein as it would do if you would have been affected with the actual virus, which just provokes a much better immune response since the protein is actually produced by the cell's own machinery, probably and possibly also carries some of the cellular modifications which are done to the protein, and it's just like a way better model of an actual infection compared to classical vaccines. So it provokes it provokes a higher immunity, and B, since you just inject the code itself, just inject the mRNA, which can be edited quite easily, you could update the vaccine for different variants. You can modify the vaccine to work against some different virus. Like you can generate a bunch of mRNAs and vaccine against everything essentially. It wouldn't work, but like not everything, but it's way easier to just edit an mRNA and create a new vaccine that way than to generate new viral protein, which you would then inject and hope that the immune response which is generated matches the immune response you would need for an actual infection.
Yeah, and it's quite interesting how these mRNA vaccines actually get into the cell, because they are basically, I think that this explains why people were so hesitant with believing that these mRNA vaccines would actually work, because for one, mRNA is extremely unstable. So that's also why you have to cool them so much, I
Yes,
think,
exactly.
to minus 70 degree. And then the other reason is that the virus is like specifically designed to bind to the cell, right? With a spike protein, but the nanoparticles, you just cover them in fat basically, and just put them into the into the muscle and hope they kind of move into the cell.
Yeah,
So
yeah.
it's a bit surprising that this works,
Yeah,
right?
well, the cell itself is a bag of fat, so a small fat droplet shouldn't have like a too
Yeah.
hard of a time to get into it. No, I'm exaggerated. It is quite complex, and a lot of the success, but also a lot of the engineering troubles and failures, as far as I know, were in designing lipid the exact lipid profile the nanoparticle needs to be able to efficiently fuse with cells and release the mRNA payload into the cell, because that's quite difficult. You need
Okay.
like the correct composition of lipids. They need to be like modified in a specific way to be able to fuse with a cellular membrane. That's really not trivial. That's very difficult. But if you nail this process, then yes, you have essentially like a mini droplet of fat and a big droplet of fat, and they just fuse to simplify it.
Okay, but that actually means that once you got that process down, then if you know how to get mRNA into the cell with this lipid, how then you can basically get anything into the cell. So then it becomes extremely universally applicable.
Yes and no. It depends on which kind of cell type you want to target. For example, for those virus vaccinations, you want to target the antigen presenting cells, which are present also in the muscle tissue. And for those, I guess I would say, unless somebody will correct us in the podcast comments, that yes, we did nail the targeting of those cells with the current lipid profile and the current nanoparticles. But if you would want to target different cells for different, say, gene therapies or something, say liver cells or I don't know, brain cells, neurons, then you would have to re-engineer them to some degree. But I'm not sure with that.
Okay. Yeah, there's also this big like range of applications on the horizon with respect to using viruses to actually treat diseases like cancer, for example. Can you? I don't know if you know too much about that, but if you do, please share it with
Yes,
us.
actually not too much. I might have to disappoint, but we're always on the search for very targeted transport vehicles of any kind, especially for cancer. That's the typical crux of cancer treatment. Initially, with chemotherapies, you just targeted essentially everything. You just gave poison and just hoped that the cancer is getting poisoned more than the rest of the body, since it's replicating so much. But yeah, people are always searching for some delivery technique or some targeting technology which is able to just target a specific cell type or a specific tumor type. And viruses are also, of course, one of those hopes. But yeah, I'm unfortunately not aware of the current state of research, and which technology is looking most promising there.
Okay. Yeah, maybe I can move it to a different podcast episode.
I would be very
from
interested in
someone from
that.
Yeah. So, I noted it. So now we've talked about viruses actually a lot. Maybe we can like move to the meta question of what your main motivation actually was to get into viruses and into this field.
Yeah, that's not an easy question because I don't think there was like one meta question. I guess it started after my masters or during my masters when I wasn't quite sure what to do with the eventual degree, and I thought about more. I thought more about the also social impact of my potential work, and I think a lot of excuse me, a lot of people in the biomedical fields see their work as, yeah, I'm helping people since I'm like researching certain diseases and so on and so forth. But having thought about like animal rights before a little bit, due to some bioethics seminar in my bachelor's where we talked about the ethics of killing and eating animals, keyword Peter Singer, which is a philosopher who did and wrote quite a lot for the animal rights movement and is generally associated with utilitarianism, the philosophy that the best course of action is the one which produces the highest amount of utility, whatever that be. I was already aware of the fact that a lot of the time there are like certain inefficiencies in the market of doing good. So some problems are like immensely overfunded, while others, which might be more important or bigger in scale, are somewhat underfunded. For example, say animal rights, given the number of animals killed for human consumption or animals killed in nature generally, compared to the amount of resources devoted to that cause. Yeah, so long story short, I read a bunch with a friend about effective altruism, which is an idea movement which focuses on the question of how to do the most good with the resources available, where resources can be anything from like, time, money, motivation, energy, on how to improve the life of or lives of the most individuals and most sentient beings as much as possible. And over that route, I was a bit disillusioned about like classical biomedical research, since the diseases are often not huge in scale, very very crowded. Like there's a lot of money going in there, and the actual impact of a single person is probably not big and might even be somewhat replaceable, since there's a large quantity of eager PhD students who really love to be paid in single digit percentages of becoming a professor eventually.
Yeah.
So yeah, I thought about more of this general question, like how to do the most good with the resources available, and really started from a blank slate, and yeah, thought about what are even humanity's biggest problems, like what do I want to do, how can I do the most good. And the effective altruism community has been very helpful and rewarding during that journey and during those discussions.
Yeah. There were already so many points in there.
I'm
I
sorry.
don't know where to start now. But that was a beautiful monologue. But I'm always hard pressed to define effective altruism besides saying it's the question of how to do altruism most effectively, which usually doesn't help people understand it any better.
Yeah.
But yeah, so you basically came up with the idea that given your biomedical background, viruses tie in nicely with questions of existential threat and biorisk. So we can first talk about that before moving to the broader questions around effective altruism.
Yeah. Yeah, for sure. So one of the directions effective altruism explored, and a lot of people went in, is the direction of existential risks or long-termism in general, like the idea that if you value happy people, then you would also value the fact that happy people would exist in the future, and the future is potentially very large. So there is some very big interest in not letting humanity go extinct. And even if you don't subscribe to that, you can still say having like 7-8 billion people go extinct is like a big, huge disaster for those 8 billion people. So quite a few people focused on reducing existential risks, which are broadly defined as risks which would, when instantiated, would lead to either the complete extinction of humanity or to a drastic and permanent reduction of humanity's long-term potential. Say like a 90% reduction of a human population would probably end civilization as we would know it, and it would be at least very debatable whether we would be able to get back to the current level where we at, or how long that would take. And of those existential risks, there are like many, some very, very present ones, like nuclear weapons and the potential of global thermonuclear war, which was probably at its height in the 60s during the Cold War. And we had a frightening number of close calls of almost nuclear wars. And if you go to the Future of Life Institute, you can see a nice, in scare quotes, nice timeline of close calls and almost disastrous events, which could very probably have led to World War III if actually going wrong. But under this banner, this header of existential risks, also fall global catastrophic biological risks, which are risks from biological systems, which would also lead to the aforementioned extinction or drastic reduction of humanity's potential. And those can be really, pandemics, broadly speaking. So it's not hard to imagine a pandemic which would lead to a significant mortality of humanity as a whole. And those admittedly very rare, but potentially extremely disastrous scenarios are just very underexplored and underfunded.
Yeah, talking about underfunding and underexplored, I think the COVID pandemic really changed our perception
Yeah, that's
in a lot
true.
of circles. And the, this morning, I actually very nicely correlated to this recording of this episode that there's an early warning center opening in Berlin, and like an early pandemic warning center. So we already offline talked a little bit about this, and you mentioned that you have looked into it, but and also read the white paper. So maybe we can quickly talk about what's happening there.
Yes, it's a new hub which is led or founded by the WHO. It's the WHO Hub for Pandemic and Epidemic Intelligence, which is now situated in Berlin at the Charité, the famous hospital where Christian Drosten is researching. So yeah, it's a hub which aims at um, which aims at synthesizing data for pandemic surveillance and then making like smart policy decisions based on those data streams. I looked, like I said, I looked briefly in the white paper. I guess anybody actually involved in that would be able to say much more because the white paper is, in my eyes, a little bit vague. But they broadly criticize that health data is very fragmented. And during pandemics, it's very hard to get like unified data sources which cover more than just whether the patient is, I don't know, male, female, and whether he died or recovered. And that like tools to work on those massive amounts of data from different countries are not really present. And they want to build a hub in Berlin where they, on the one hand, collect and curate a bunch of different data relevant for epidemics and pandemics, and then build tools for predicting certain outcomes, and also foster collaboration and subsequent policy making based on those insights. And it's looking pretty interesting. They have, I think, 100 million euros initial funding. So there's definitely stuff happening in that sector. But yeah, I think it remains to be seen what they will actually do and implement and how people will react to that and whether it's long-term viable. Because I do see various such projects popping up as some sort of, some sort of just blind reaction to the current situation without having actual long-term potential. And if we take another, I don't know, 50, 60 years until the next pandemic comes, then for me at least now, it's not clear whether those institutions will still be funded and have money and are present in the first place. Whether it's just like a hype bust cycle.
There are also elections coming up
Yes.
in these democratic countries.
Exactly. And why fund like an expensive hub if you didn't have a pandemic for the last 50 years? Yeah.
Yeah, exactly. And with the incentive structure of democracies usually being on the scale of 2 years, like of the time scale, then
Yeah.
usually long-termism gets ignored, or at least like a lot of stuff is swept under the
Yeah,
rug.
benign AI autocracy soon.
Yeah. Yeah, let's hope for the best. But talking about like a lot of money flowing into maybe also questionable directions at the moment, I think the biochemical or the virology community is also a bit torn on the question of whether we should actually spend more money in investigating these kind of viruses and how they transmit to human beings. And this whole expression of gain of function research has been thrown around in the media quite a lot. And there was also this famous like showdown in the Congress between Fauci and Rand Paul, which also centered around his involvement in gain of function research and the potential question of the lab leak that is still unresolved, and the idea that maybe we basically mankind brought this down upon themselves, and we actually the man-made pandemics are the biggest risks. I don't know how you stand on that. And we have also offline already talked a little bit about your stance on gain of function research, so maybe we can delve into this in a little bit more detail.
Yeah. So first of all, I do think that like engineer pandemic pose the greater risk just because engineering is able to explore and design the, say, sequence and fitness space much faster and much more directed than evolution can, and might be able to jump some gaps which are potentially not jumpable by like stupid blind selection pressures. And especially if some sort of malice is involved, then it's usually easier to exploit known weaknesses than if you just have some natural pandemic. But that just as a broad disclaimer, that I do think for the tail risks, people should focus on engineered pandemics and maybe also look into and specifically work on countermeasures, which might be able to intercept engineered pandemics. And there's also some amount of community norms present, like how should we deal with info hazards and how should we like monitor ourselves, which I alluded to in the beginning of the conversation. In terms of gain of function research and like more broadly dual use research of concern, so research which has like a dual use nature, which is on the one hand beneficial for some purpose, but on the other hand also generates some downsides, like be it potentially directly negative applications of that technology or some generation of knowledge which might be able to be misused in the future. I don't think this is getting discussed enough. So this is definitely, in my eyes, a very much underrepresented topic. And virology might be one of the fields where this is maybe most neglected, because I think the synthetic biology field has embraced this a bit more, especially if you look at the iGEM, this yearly competition for undergrad teams in synthetic biology. They now have a dedicated biosecurity seminars and a biosecurity person, and they are nudged and encouraged to think about those topics. But in virology, there's often still this notion of, it's a scientific question, so it's positive if we research it, or there are viruses out there, so it's positive if we go out and collect them, without really thinking about the possible downsides of such projects. And gain of function research, where you give a virus additional capabilities or modify additional capabilities of the virus to find out some key facts about its biology. They are one side of that, but also just virus collection, going out into nature to, say, some bat cave and sampling bats and exposing yourself to the bats to see what kind of viruses they have. There are things there are possible upsides to this kind of research, but there are also like drastic possible downsides to that research, and I don't think they are discussed enough.
I think it was even a point you mentioned when we last spoke, was about the fact that a similar coronavirus was found by the Chinese authorities, I think in 2009 or something, in a cave, and then it was brought back to the Wuhan lab. And people argue that it makes sense to do this kind of research because then we were already familiar with similar bad coronaviruses. But what actually came out of us knowing about this virus was basically zero. So we didn't have any antiviral medications. Some people have tried to develop vaccines, but they also never got into clinical trials. And so basically, even though we did research on viruses where it's still argued that potentially could have led to a lab leak that then infected or like brought this whole pandemic upon us, but the insights we gained were basically zero still.
Yeah, that's a good point. Like you said, I'm not really sure that we gained that much from, or anything from previous research on based in that direction. And I think most of the benefits we got from this for ending this pandemic were essentially in mRNA vaccines. The mRNA vaccines were luckily just at that point in time where they were ready to be employed widely. They were designed really quickly. I think like a week after the virus genome was published in January 2020, the mRNA virus design was finalized, well, not finalized maybe, but was penned, and yeah, it took just a year, well, still a year, of regulations and so on and so forth, and we had a vaccine. So that's like a huge boon. But like in terms of concrete coronavirus virology, I don't think it really prepared us for this pandemic. Like, yeah, we had some previous knowledge about some biology of the virus, where and how it replicates, but this was, as far as I know, mostly gained from the SARS-CoV-1 epidemic, and not really from additional virus sampling work done in the subsequent years.
Yeah. What do you think are the best ways to prepare for future pandemics? Is it more monitoring or like more biosecurity? Or how would we actually deal with this?
Yeah, so one very important approach is just monitoring. People like dream a bit of like widespread biosurveillance and widespread environmental monitoring, where you essentially sample and sequence parts of the environment, say wastewater or air at airports constantly, and check for any kind of sequence which you either recognize or don't recognize, but it's like spreading exponentially or something, to really quickly identify viruses. And also less science fictiony, just sequencing in hospitals, like more widespread metagenomic sequencing in hospitals, that if you have a patient which comes into your hospital or comes into the doctor's office and he has some symptoms which are not explained by the usual palette of tests against the typical common cold or influenza viruses, that you just immediately do a metagenomic sequencing of his I don't know, sputum or blood or whatever material you have, and check for unknown viruses. And this is coming. Like, this is not super science fiction. Hospitals are rolling this out more and more, usually for more difficult and more severe cases. If a patient is in, say, the intensive unit with some, say, encephalitis, and you don't know the etiology why he has encephalitis, then this is already done, but not routinely. And I think we would greatly benefit for this, not only for pandemic preparedness, but like generally for knowing what's going on, like what kind of viral diseases are even in our population, because we just don't know most of the time. Like, people say, yeah, I have a common cold, but what exactly? Why? Like, how's the dynamic of this specific virus? We just don't know.
Yeah. Also nicely illustrated by the fact that I think I was actually surprised last year when naturally everyone was talking more about viruses, to hear about the fact that there's actually 100 different viruses that are all responsible for causing what we just very vaguely label the common cold. So like no one really cares about differentiating in any kind of way. And then you have the flu, and there's also only one word for the flu, but no one really understands that there's so many different dynamics going on. So one idea that connects to this is what Michael Mina proposed in a podcast I recently listened to. Something I think that's also being discussed, something similar to a weather forecast for viruses, because we are all used to just checking the weather forecast when we go out, like to just check for the next week what we could expect. And if you introduce something similar for viruses, we can, for example, if we have like a good idea, as we do with the coronavirus right now, in which area of the country, for example, in Germany, we have a certain prevalence of a certain virus, then you can introduce mask wearing when it's actually necessary, because then you can also avoid like influenza pandemics, or like the flu is not something nice to have. Most respiratory viruses are also not, it's not nice to have them, even though they won't kill you as much as the current coronavirus does. But yeah, to just introduce something, this kind of idea, and then you just spend 1 or 2 weeks in winter wearing a mask when you know in your area of the country you have a lot of a certain virus. Then you just prepare for that, and then it's going away again, and basically, yeah, much smarter about it.
Yeah, that's a pretty nice idea. I like that. It will probably be some sort of trade-off because a certain amount of like pathogen exposure for the immune system is also healthy and necessary, so it doesn't get bored and start reacting to pollen or house mites or whatever.
Yeah.
But yeah, for example, in the flu season, definitely that I would be all for that. But same for environmental monitoring. The technology is not quite yet there yet. Sequencing is getting cheaper and cheaper, quite drastically so, but not cheap enough and not easy enough that it can just be done everywhere all the time. And also the bioinformatics and computational pipelines are not up to the task right now. But given that Moore's law is still going somewhat strong, unless we have the chip shortage for a few more years, I'm
Yeah.
more optimistic that we actually will see more widespread sequencing and surveillance starting from hospitals than coming to probably more wealthier medical centers, but eventually maybe at every residential doctor's office. Yeah.
So how much is sequencing, like a current, for example, the coronavirus, like as a ballpark figure?
So it's less than 100, more than 10. I would say it depends on the exact technique you're using. So there are like multiple sequencing technologies in use, and also it depends on how many samples you sequence in parallel, and that might differ quite drastically between, say, a very small hospital versus a big genomics center where they can really dig into the economies of scale. But it's not super expensive anymore. Like it's cheap enough that most hospitals can do it for research purposes. But it's still too expensive to do it regularly for just every sample, given that a PCR, which checks for the typical respiratory panel, costs just a few bucks.
So now we've already spent 90 minutes talking about viruses.
Yes.
We got drawn back into it.
I mean, there are a lot
I
of
wanted
viruses,
to
to be fair.
address, yeah, yeah, I wanted to address some of the other EA related topics because I also think they are very interesting. But yeah, in the interest of time, maybe we need to focus a little bit on what's most interesting. Do you have a favorite EA related topic besides what we've already talked about?
That's hard to answer really, since effective altruism is quite broad in its interests. Like the general question how to do the most good is perhaps on the surface quite easy, since you can maybe naively say, okay, what are the world's biggest problems? Okay, so we have, I don't know, pandemics, we have nuclear war, we have global poverty, we have animal welfare, we have climate change, we have maybe risk from unaligned artificial intelligence, and then which problem is the most easiest to work on, so which is like the most tractable problem, and which is the most neglected problem, and then just try to you know find your solution to these questions and work on that. But the thing is that the world is pretty complicated, and there are a lot of topics which you ideally need to understand to be able to approximate those assessments regarding your scale, tractability, and neglectedness of problems. And yeah, there's also the potential that you might be missing some ongoing moral catastrophe that you are actually not aware of. Say, a couple of 100 years ago, people didn't really think that animals were like moral patients deserving of some sort of like consideration. And up until not like super recently, people were also performing surgeries on newborns without anesthesia because they're thinking that newborns can't feel pain. Turns out they very likely can, and that was a bad idea. But there might be something which we are just missing because it's not in our current paradigm of moral consideration. And so rather than, I don't know, digress more into like topics like underlined artificial intelligence, which you should definitely do a podcast episode about.
Yeah, I already have too many AI podcasts, so we don't need to
Okay.
talk about that.
But AI risk
Yeah.
podcasts. I guess one topic I really like, which is still underexplored by a lot, I would say, is wild animal suffering. The notion that nature is quite cruel, that the average animal in nature in the wild is probably suffering and living a life which might be negative on net, and that this is actually something which we want to work against or do something about, which does provoke quite drastic emotional responses in some people. But yeah, maybe you have questions about that.
Yeah, I just wanted to mention, like, I think it's also an expression by Peter Singer that I find quite nice. This idea of the circle of concern that is widening across, like, as we morally, like, arguably, as we make moral progress, if you can say that. Some people argue you can't, but from a moral objectivist perspective, there's some claim to be made that the suffering of conscious beings is something to be avoided. And as we progress in our understanding of neuroscience, for example, and then we are slowly starting to realize that not only our tribe or our race or the human population of Earth is relevant, but now we can also add all the animals that we can understand have conscious content if in some way morality is really as deeply intertwined with conscious content as we currently think, or I think many people hold it to be true. And then we, yeah, can come to this idea in the terms of wildlife. One reframing I found very nice is to say animals are living in extreme poverty, because it's something that makes sense to us from an animal perspective. Yeah, like from a human being perspective, because nature is, as you say, pretty cruel, and we associate poverty with lack of resources, constant threat. You don't have you fight with a lot of viruses and disease, and you don't, like, childbirth is pretty dangerous, and all these kind of things that human beings are slowly distancing themselves from, especially in the first world. And then if you come, like make the connection to wildlife, then you have a lot of suffering popping up there suddenly that we haven't really dealt with. And I was really, as you mentioned, it brings out a lot of emotions. I remember the first time I heard about this whole approach, it always brings up this idea of, are we kind of overstepping our reach if we try to make nature, like remake nature, and try to claim that nature is inherently cruel and we need to, as men, come in and kind of save it from itself. It's kind of like a savior complex or something. There are also good claims to be made, so maybe I can hand back to you, and so
Yeah, that's
you
true.
can elaborate on your position. Yeah.
Yeah. People definitely do have that reaction, and they might just assume that this position implies that we should, I don't know, start to fix all of nature, just go out there and I don't know, murder all big charismatic predators and apex species, which is of course not what any sensible person is advocating for. But yeah, I would counter that like on one hand, we are already interfering in nature quite a bit, like quite drastically, without any concern of welfare. Oftentimes with just the opposite of welfare as a direct result, just given the domestication and factory farming of species, or burning down acres of rainforests for just say soy plantation, also for livestock feeding. Although I could be now very contrarian and say that maybe burning down the rainforest for soy plantation might be positive for a wild animal suffering perspective, since since an acre of soy plantation is able to, or is less biodiverse and has less suffering per acre than rainforest. I'm not going there though. But for example, here in Germany, we essentially vaccinated foxes and wild dogs against rabies, which we primarily did for ourselves, since we as humans didn't want to get rabies. But I'm sure the fox population was also, like, the foxes were also quite happy that they didn't have rabies. They weren't aware of it, of course, but on net, it probably increased welfare in the fox population. And rather than just going out and burning down all forests, or sterilizing all predators, there are definitely approaches you can take, say vaccinations or birth control for some animals which are at risk of overpopulation and starvation, for example. Like, would you rather have a population of animals populate like crazy and then have 90% of them starve to death, which is quite unpleasant, and end up with some ecosystem with 10% of the animals, or just give 90% of them birth control via like feeding, and end up essentially with the same ecosystem and 10% of the animals? Or the question of whether we want to bring back predator species like wolves into ecosystems where now animals are like not chased to death and eaten alive. That's that's also a question which, for me, does fall under this question of, or under this consideration of wild animal suffering, but is quite reasonable to discuss, and people discuss that, and it's not super out there. So there are things you could do to work on this topic, which don't entail like drastic changes of ecosystems. Also, just research on the topic. Like, yeah, if we don't know the consequences of our actions and what's out there, then like researching it might be a great idea. And just because we're saying like, no, it can't be done, it's too complex, we don't know our actions, that doesn't mean that that this will be the case forever, and that we therefore mustn't do research in that direction.
Yeah, that's a very good point. Yeah, and I like the rabies example because it's, yeah, very concrete way of understanding, like a lot of the diseases, as you mentioned, the coronavirus might come from bats, and actually in the Arabic world, you have this MERS virus, which usually comes from camels, and it probably also came from bats. But then it actually makes more sense to not give a MERS vaccine to hundreds of millions of people living there, but to the couple of camels that are kind of at risk. Or in the example of rabies, it's also very expensive to give rabies vaccine to everyone. But if you can, especially because right now in this area of the world, we don't have too many animals at risk, too many foxes, and the dogs are usually domesticated enough and not running around in the wild. Yeah. But I guess like from an animal welfare perspective, we also have a bit of like more obvious targets with the way we deal with animals in the like meat production sector and dairy production sector. Are you also interested in that topic? Or?
Yeah. Like I said at some point during the interview, I read Peter Singer as an undergrad, and was pretty convinced by his arguments. And the factory farming and mass production of animals and fishing as well is probably like the cruelest thing that humans are currently doing, and probably also the biggest source of suffering, which is at least easily influenceable by humanity. I'm not sure whether wild animal suffering is smaller than suffering in factory farms from a like total scale. And there are some assumptions you have to make for that. But yeah, factory farming is just like very obviously immensely negative for the animals involved, for the environment, for the climate, for the people themselves, given that most animal products are not hugely healthy, and that a lot of the factory farms are breeding grounds for zoonoses and potential future pandemic viruses, for example, swine flu, avian flu, and also antibiotic resistant bacteria, which are very, very on the rise, very much on the rise, given the just sheer abundance of antibiotics used in factory farming. So I see a huge, huge number of externalities, of negative externalities, with almost zero upside given the quality of meat substitutes we currently have, and we saw in the last years. Yeah.
Yeah, with a limitation to I guess Germany. Maybe I was in vacation in Spain, and the concept of vegetarianism isn't as widely spread there. I don't have the
Yeah.
numbers, but it's usually there. They don't really even have any vegetarian alternatives, and sometimes don't even know the concept if you try to explain it to them.
Yeah,
So
yeah, of course it's a very cultural and of course also like developmentally different situation, in a lot of d- other countries, compared to say Central Europe and the US, where we have this upside, this benefit of good alternatives, but I see this less as a like fundamental challenge to the cause, but rather just a lacking behind in time, since also a lot of other countries are trending towards more like moral consideration of animals and subsequent changes in the food production and availability of alternatives. I think it's very rare to see like a fundamental cultural opposition to such things.
yeah, that's very true. Yeah, I think that the trend seems very strongly heading into one direction, and it's I don't think it's really a controversial topic. I think no one really likes animal suffering, or like it's not a significant enough percentage of humans to that they would have any political saying in that. It's just mostly pragmatics and like bad incentives, mostly financial incentives in these industries. But if there's enough pressure, like consumer pressure and the willingness of people to pay a bit more, then I think it's going to happen pretty much on its own. And I mean, it seems that it's already happening. Like all of the big German supermarkets already have a lot of alternatives and like eggs like from biological production and milk as well. So it seems it's getting much better.
Yeah. I mean, for milk and cheese products, I see that it will take probably the longest. Well, meat you can pretty easily emulate with plant protein. For cheese, yeah, we will probably have to wait for lab grown milk or lab produced milk, based on yeast cultures or something, for us to see some big change there. But at least for meat, I'm more optimistic.
Yeah, that's true. And there's also synthetic meat on the
Yes,
horizon, maybe even at reasonable prices.
hopefully.
So considering it's getting close to the 2 hour mark, I wanted to to close with some like also meta questions. Maybe on one question I like to ask is whether you have a favorite book or like a book that already like maybe influenced you the most intellectually speaking. You already mentioned Peter Singer. Maybe this is one of them, but also other ideas, options, also recommendations. Something you're reading right now.
Right now, I'm reading mostly science fiction short stories. So they are fun to read, but don't really influence my thinking that much. Um, for a book, probably Super Forecasting by Phil Tetlock. That really changed my thinking quite a bit, and more into the probabilistic direction. So it deals about making like good forecasts and how to think, how to think well in general. Um, One other book I guess really changed a lot for me was Rationality from AI to Zombies, which is a series of online essays written by Eliezer Yudkowsky, an AI safety researcher, condensed into book form. It's pretty long, a pretty hefty read, but yeah, it's just very insightful, and as the title suggests, it deals with human rationality, which is a fascinating topic.
Yeah, Yudkowsky is a very fascinating guy and
He is.
good writer as well. Yeah.
Yes, very much.
What's his? He also has his. Is it called Applied Rationality or like his website?
There's this, the Center for Applied Rationality, CFAR, which gives rationality workshops, and the rationality from AI to zombies are based on essays he posted on Less Wrong, which is a community of people discussing human rationality and trying to enhance it.
Yeah, I was wondering if we should quickly get into the effective giving topic, but maybe it's we don't have enough time to cover that to any satisfaction. Um, yeah, maybe we can quickly talk about 80,000 hours. I think you also mentioned using that resource to
Mhm.
decide on your career, and I also used it quite extensively and found it
Yeah,
extremely insightful and helpful and like gaining a new perspective on your life. Yeah.
yeah, definitely. Like I said, the question of effective altruism, how to do the most good, given the resources, does not all only include money, which we now didn't discuss, but also time, and the time spent on your career, on your job, is perhaps the most time you spend on anything in your life. It's roughly 80,000 hours, hence the name of the organization. And 80,000 hours gives career advice for altruistically motivated people on how they can maximize the good they do with what they do in their future professional life. And 80,000 hours does coachings. So you can register for a one-on-one coaching with one of their career coaches, which I found very helpful. But they also have a website with a wide variety of essays and topics and reports on different problem areas like factory farming we talked about, or biological risks we also talked about, nuclear weapons, global poverty, artificial intelligence, and so on and so forth, where you can really go in depth and just explore different cause areas and problem areas to see where you could potentially work. And they have a wide variety of resources on like general career advice as well. So yeah, I highly recommend checking out 80,000 hours. Yeah,
Yeah,
great
I
website.
very much agree. Now that church bells are also going off here. Yeah,
Yeah,
so maybe
here as
with
well.
this beautiful, I think they are all in the same key, so it won't be too bad of an experience for our listeners. lasted until this point of the
Yes.
conversation. So I think now it's time to wrap it.
So now
Yeah,
to church,
now it's
everybody.
time to wrap it up and go to church. After this was the second
Exactly.
longest conversation we had so far. So I feel like we still have too many things to talk about, but it was a super interesting conversation.
Yeah.
Mhm.
Thanks. I
Yeah,
really enjoyed it. Very, very good questions. Very, very in-depth
yeah.
conversation. I really
I
liked it.
hope other people will like it as well. And thanks again.
Yeah. Thank you very much.